I am Dr. T Elakkiya, Assistant Professor, PG and Research Department of Chemistry, Sri Sharda College for Women, Autonomous, Salem 16. Today's topic is thin layer chromatography. Introduction to chromatographic techniques. Chromatography is a powerful analytical technique used to separate, identify, and quantify components in complex mixtures. These techniques rely on the principles of differential migration and positioning of analytes between a stationary phase and a mobile phase, allowing for the precise separation and analysis of even the most intricate chemical compositions. From pharmaceutical development to environmental monitoring, chromatography has become an indispensable tool in various scientific and industrial applications, providing unparalleled insights and enabling groundbreaking discoveries. So, what is meant by thin layer chromatography? Yes, thin layer chromatography, we also called as TLC, is a versatile analytical technique used to separate and identify components in a mixture. It involves the separation of compounds based on their relative affinities for a stationary phase and a mobile phase. Principles of thin layer chromatography. In thin layer chromatography, some adsorbents like silica gel, alumina, etc. are supported as thin layers on glass plates. We also called as chromatoplates. This technique is similar in some aspects to both column and paper chromatography. Just as in column chromatography, the moving substance is attracted by the polar sides on the surface of the adsorbent by electrostatic forces. This binding is reversible. There is a threefold interaction between the solvent and the adsorbent, the solvent and the compound and the adsorbent. So we have a wider choice of the media, so we can separate compounds by partition, adsorption, and ion exchange. Adsorption. Compounds interact with the stationary phase based on their affinity. Migration. The mobile phase carries compounds at different rates based on their properties. Separation. Compounds are separated into distinct bands or spots on the TLC plates. Choice of adsorbent: silica gel, alumina, kieselguhr, cellulose are used as adsorbent material. There are a number of materials which are used as adsorbents. First, silica gel. This is the most widely used. This is used along with a binding agent like plaster of Paris. Alumina. This is basic in nature. It may be applied with or without plaster of Paris. Hazelgar. This is neutral in nature. Its absorbing capacity is less than either silica gel or alumina. It may be applied with or without plaster of Paris. Cellulose. Several cellulose powders with a variety of ion exchange properties are available. They may be applied with or without plaster of Paris. Commercial adsorbents. Commercially, lot of adsorbents are used. Polyamide powder, calcium sulfate, magnesium silicate, powdered glass. Instrumentation and equipment. TLC plates. TLC plates coated with a thin layer of silica gel, alumina or other adsorbent. Then second one, development chambers. Sealed chambers that hold the mobile phase and allow for controlled development. Visualization tools, UV lamps, staining reagents and densitometers to detect and quantify compounds. Sample preparation. First, we want to dissolve the sample in a suitable solvent and apply the sample as a small spot or band on the TLC plate. Then allow the spot to dry completely before development. Then optimize the spot size for best separation. So first, we go to choice of solvent. Choice of solvent. So the choice of solvent depends on the nature of the substances to be separated and on this adsorbent used. Generally, the polarity of the solvent and the substance is matched, and then the choice of solvent is made. More polar solvents produce greater migration and this gives better separation. The choice of solvent should be such that the position of the compound must be halfway between its point of application and the solvent front. Combination of two solvents always gives good separation than a single solvent. The choice is usually made from the petroleum ether, carbon tetrachloride, benzene, pyridine, acetone, water, etc. Then next one, preparation of chromatogram. That is we called as chromatoplate. Square or rectangular glass plates with the sizes ranging from 2.5 into 20 centimeter to 20 into 20 centimeter are used as adsorbent supports in TLC. Plastic and metal foils can also be used instead of glass plates. The adsorbent is taken in a that's a mostly the most widely used adsorbent is silica gel. A weighed amount of the adsorbent is taken in a bottle. Water is added to it. The bottle is stirred or shaken vigorously until we get a homogeneous, thick, mobile slurry. Two plates are put together. They are held by the thumb and forefinger at one end. Now they are dipped in the slurry, taken out and or held vertically. The solvent dries up. The dry plates are separated. Now we get two chromatoplates. There are also other apparatus for the preparation of chromatoplates. Then next one, activation. TLC involves adsorption. So water or other polar solvents affect the development. So they must be removed from the chromatoplates. This is known as activation. This is done by drying the plates in an oven at 100 to 150 degrees Celsius for about 2 hours. Sample application. A small amount of the sample is dissolved in a small volume of a volatile solvent such as benzene, ether or ethanol. The choice of solvent depends on the nature of the substance to be separated and the nature of the adsorbent. For polar substances, polar solvents are used. Less polar substances should be dissolved in a suitable non-aqueous solvent. Activated silica gel or alumina should be used as the adsorbent. A baseline is drawn about 2.5 centimeter from one edge of the plate. The samples are applied on the small spots 1 centimeter apart on the baseline. The solvent is evaporated. Solutions of standard substances are also applied by the side of the test samples. Next one, development of the chromatogram. The chromatogram is usually developed by ascending method in a developing chamber called tank. The atmosphere in the tank is saturated with the solvent paper by placing a paper impregnated with the solvent around the side of the tank. The chromatoplate is placed in the tank and the tank is filled with the solvent to a depth of about 1 centimeter. The tank is closed firmly with a lid. After a certain time, when the solvent has moved to about 10 to 20 centimeter above the origin, the plate is removed from the tank and the solvent front is carefully marked. The solvent is evaporated. Then last one, location of compound on the chromatogram. Colored compounds can be identified by visual inspection. To identify colorless compounds, physical or chemical methods are adopted. Water is sprayed on the chromatogram. Hydrophobic compounds show their presence as optically dense waxy areas. The developed chromatogram is inspected under UV light in a darkened box. Many compounds appear as dark spots in the light background. Crystals of iodine are placed in the tank during development. It imparts a dark brown color to the spots. The sample is mixed with a small amount of a radioactive isotope and applied on the plate. The radioactivity is measured by a Ziegler-Muller counter after development. They may be detected by audiography, autoradiography also. So this is optimizing solvent composition, solvent polarity, solvent strength, solvent ratio. Adjust the polarity of the solvent system to target the specific compounds of interest. Solvent strength vary the solvent strength to control the distance the compounds travel on the TLC plate. Solvent ratio, experiment with different ratios of solvents to find out optimize the for separation. And we are already seen that developing the chromatogram, that is solvent application, chamber development, compound visualization, and also so on. Then interpreting TLC results. Interpreting. That is very important. It is retention factor, that spot shape, spot intensity. That is retention factor, the distance a compound travels relative to the solvent front used to identify compounds. The shape of the compound spot can indicate purity or potential problems in the separation. The intensity of the spot can provide information about the concentration of the compound. So first we are understanding retention factor values. What is retention factor? Yes, the RF. Retention factor value is a dimensionless quantity that represents the ratio of the distance traveled by a compound to the distance traveled by the solvent front on the TLC plate. Range of RF values range from 0 to 1, where 0 indicates a compound remained at the baseline and 1 indicates the compound traveled the same distance as the solvent front. Factors affecting RF values are influenced by the nature of the compound, the mobile phase, the adsorbent material, and experimental conditions such as temperature and humidity. So how do we calculate the RF values? So RF value is calculated as RF is equal to distance traveled by the compound divided by distance traveled by the solvent front. Yes. First example, we see if a compound travels 3 cm and the solvent front travels 6 cm, the RF value would be 3 cm divided by 6 cm is equal to 0.5. If a compound travels 1 cm and the solvent front travels 5 cm, the RF value would be 1 cm divided by 5 cm is equal to 0.2. So we easily calculate the RF values. So how we interpret RF values in TLC analysis? Identification. Second, purity assessment. Reaction monitoring. In sample comparison. So identification. The RF value can be used to identify unknown compounds by comparing them to the RF values of known standards. Reaction monitoring. Changes in RF values can be used to monitor the progress of a chemical reaction or the separation of reaction products. Purity assessment. The presence of multiple spots on the TLC plate indicates the presence of multiple components which can help assess the purity of a sample. Sample comparison. Comparing the RF values of samples can help identify differences in their chemical composition or impurities. So here we need separation and visualization. That is development time. Control the development time for optimal separation. Use UV lamps to detect fluorescent or absorbing compounds. Staining. Apply specific stains to detect and quantify separated compounds. Densitometry. Measure the intensity of bands to quantify compound amounts. So, applications of thin layer chromatography. Thin layer chromatography is a versatile analytical technique used to separate, identify, and purify a wide range of chemical compounds. It has numerous applications across various industries from pharmaceutical to environmental analysis. qualitative analysis of compounds, identification of unknown compounds, purity determination, monitoring reactions. TLC can be used to unknown compounds by comparing their retention factors to those of known standards. Purity determination. TLC can assess the purity of a compound by detecting the presence of impurities or byproducts. Monitoring reactions. TLC can be used to follow the progress of a chemical reaction by detecting the formation of products and the consumption of reactants. Separation and identification of drugs. Pharmaceutical analysis, forensic applications, clinical diagnostics. TLC is widely used in the pharmaceutical industry to analyze and identify drug compounds as well as to monitor drug quality and purity. TLC is a valuable tool for the identification and separation of illicit drugs and other substances in forensic investigations. TLC can be used to detect and quantify drugs and metabolites in biological samples such as blood or urine for clinical diagnostic purposes. Monitoring organic reactions, reaction progress, second one reaction optimization, then reaction completion. TLC can be used to monitor the progress of organic reactions by tracking the consumption of reactants and the formation of products. TLC helps researchers to optimize reaction conditions by allowing them to quickly and easily analyze the reaction mixture at different time points. TLC can determine when a reaction is complete by observing the disappearance of starting materials and the presence of only the desired product. Analysis of pigments and dyes. Art and archaeology. TLC is used to analyze and identify pigments and dyes in workshop art, archaeological artifacts, and historical textiles. TLC is employed to ensure the quality and purity of natural and synthetic colorants used in food, cosmetics, and personal care products. TLC can detect and quantify environmental pollutants such as pesticides and industrial dyes in soil, water, and air samples. TLC is used to identify, quantify, and control the quality of dyes and pigments used in the textile manufacturing process. Then it is also used as purification of natural products. TLC is used to isolate and purify bioactive compounds from medicinal plants for further research and drug development. TLC helps analyze and purify the complex mixtures of volatile compounds found in essential oils. TLC is employed to extract, separate, and identify novel compounds from marine organisms such as algae and sponges. TLC is used to isolate and purify biologically active compounds from fungi, including antibiotics and other pharmaceutically relevant substances. Quality control in the pharmaceutical industry. Raw material testing ensure the identity and purity of starting materials used in drug manufacturing. Batch analysis verify the consistency and quality of drug products across different production batches. Stability testing monitor the degradation of drug substances and formulations over time to ensure product integrity. Impurity profiling identify and quantify any potential impurities or contaminants in pharmaceutical preparations. Then, future advancements in TLC techniques. Automation, that is, developments in robotic sample application and image analysis will streamline TLC workflows and increase throughput. Coupling TLC with analytical instruments like mass spectrometry will enhance identification, quantification, and capabilities. Microfluidic TLC platforms will reduce sample and solvent requirements, making the technique more environmentally friendly. Advances in data processing and machine learning will enable more accurate automated interpretation of TLC results. We have some quiz questions now. So now we clearly and thoroughly know about TLC, that is thin layer chromatography, principles, then choice of solvent, choice of adsorbents, how we develop the chromatogram, how we interpret the results, what is meant by RF value, or then its applications also. Now we just see the quiz, just simple 10 questions. First question, what is the primary purpose of thin layer chromatography? So the answer is separation and identification of compounds. What type of stationary phase is most commonly used in TLC? Most commonly used in TLC is the silica gel. In TLC, what is the mobile phase? The solvent or solvent mixture that moves up the plate. Which of the following is not a common method of visualizing spots on a TLC plate? UV? No, we use UV. Iodine vapor? Yes, of course we use iodine vapor. Ninhydrin spray? Yes, we are using ninhydrin spray. Then other one is X-ray diffraction. So the answer is X-ray diffraction. X-ray diffraction technique is not a common method of visualizing spots on TLC plates. What does the RF value in TLC stand for? That is retention factor. How is the RF value calculated in TLC? Distance traveled by the compound divided by the distance traveled by the solvent rate. Seventh one. What is the main reason for using a developing chamber in TLC? To saturate the atmosphere with solvent vapor is the correct one. What could be reason for the poor separation of compounds on a TLC plate? Too high concentration of the sample, using a stationary phase that is too polar, inadequate development time, all of the above. Yes, all of the above. Too high concentration of the sample, using a stationary phase that is too polar, inadequate development time are the reason for the poor separation of compounds on the TLC plate. Which of the following is true about the polarity of solvents used in TLC? The polarity of the solvent can affect the RF values of the compounds. What is the primary principle behind chromatography? That is differential solubility to phases. Thank you.